So, cops and probably a detective, a lady in hair with hair curlers, bunch of dead cats. Notice all the computations, theoretical scribblings, and lab equipment, Norm. Yes, curiosity killed these cats. But you gotta be curious. You gotta wonder what's going on and how to test it. So we're working our way through issues with design. And one of the things you gotta take into account are confounding factors. These are confounding factors are things that are going to mess with your results that you're not actually testing. And you don't want them there. Sometimes you can't help but include them. And sometimes you can design around them. Is that there's a lot of different confounding factors. But one really good example that I said I would come back to is the confounding factor that you have when you run tests on people, especially drug tests on people, is that what are you going to measure? Sometimes you can measure very specific things. With hydroxychloroquine, you can measure symptoms. How do you measure symptoms? Not all symptoms are easily measured. You may say to people, so, how much coughing have you been doing? What's your temperature been at these times of day? That you can measure with numbers. But, how do you feel? That's a pretty subjective measure. And just the act of being treated for something, people are going to tend to feel better. That's a confounding factor. I'll tell you, one of the early examples of confounding factors is that when people started to use mice as models for drug tests and stuff. So you'd have you need mice that get whatever illness you're testing on. And you have your experimental mice that you're going to treat, and your control mice that you're going to compare them to. And so they're, testing, they're doing, say, injecting the mice with something. And some bright person in the lab went, so we're grabbing these mice and we're sticking needles in. Yeah. So does that have an impact on whether they get better or how quickly they get better? I don't know. Maybe it does. We're not doing that to control mice. Oh, we're not treating them the same. The only thing that should be different between your two tests is the variable. And if you're performing a test on one group and you're not doing anything to the second group, you're not really giving comparable tests. So they needed to come up with something they could inject the control mice with that gives them the experience of the injection but not the testing of the variable, the drug itself. When you test on people, if you're going to inject people in your experimental group, you got to inject the people in your control group with a salt solution, something that you're sure it's not going to have any effect on the condition that you're testing, but will mimic the experience. You're going to give a pill to this group, you've got to give a pill to that group. Because what you need, now this is not a confounding factor, this is what you do to work around the confounding factor. You administer a placebo. There's what they call the placebo effect, is that people who've been treated have results that are different than people that haven't been treated. For a vaccine, for instance, then the placebo is not as important because you're measuring the ability to actually pick up and support a virus or whatever it is that you're testing. So it's good to have a placebo group, but you don't necessarily need to be giving them, the same treatment. But somebody could come along and go, but what if just the act of giving them a shot boosts their immune response, and so they're not as likely to get, you go, okay, I guess we need a placebo group to get in a fake vaccine injection. But then you run into the ethics, which is another confounding factor of if this is a dangerous disease and we could be giving them protection, and we're pretty sure that the vaccine isn't going to be bad for them, then not giving everybody the vaccine may not be ethical. And I suppose I should write that down as a confounding factor. There's all sorts of effects. In the case of placebos, we use what's called blinding. Used to be they would give the drug to the test group and they would just compare it to people that they weren't even using, they were just sick. How does these people getting better, the sick people are not getting better, you know? Then they realized that they were ignoring a possible placebo effect, so everybody got treated. And that's single blind. Single blinded is you've got two groups, and as far as they're concerned, they're all getting the same treatment. Then they realized that maybe the people giving the treatments out weren't treating the two groups equally. And it turns out somebody tested that and found out that doctors treating the placebo group were more likely to talk about things like getting worse because they weren't expecting the placebo to do anything for the condition. And people that were administering to the test group were more likely to talk about side effects because they knew they were actually getting a drug that would have side effects. So they do what's called double blinding now, where if you're in a double-blind study, you don't know which treatment you're getting, but the person giving it to you doesn't know either. So they can't inadvertently, you know, treat you one way or another, treat you differently, because they don't know. They don't know if you're in the control group or the test group. They're thinking, in fact, they've done this in some studies, of adding a third level, what they call triple blind, because so much of modern drug discovery is there's a lot of math involved in comparing groups and this and that and this parameter and that parameter, and a lot of times the data's got to go through number crunchers, you know, bioinformatics specialists. And somebody came up with the question of, it seems like math, but there are ways to treat the numbers that are somewhat subjective. Does it matter if the bioinformatics people know which of the two groups' numbers they're doing, the test group versus the placebo group? And somebody went, well, why don't we just not tell them? Do this group, do that group, we're not telling you which one is which. That's triple blinded. So single is the patients, double is the people doing the treating of the patients. All the treatments come from the drug company, just numbered and administered that way. So the drug company knows, but they're not, you know, in the room with the people. And then triple is, sorry about that. I had it turned off until yesterday when my son said, you gotta turn it on so I can get a hold of you. And I should have turned it off before I did the video. Anyway, so ethics is a confounding factor. Placebo effect is a confounding factor. Placebo effect is very powerful when you're doing tests on things like herbal remedies. You find that when you do a comparison between people actually getting the herbal remedies and people getting a fake herbal remedy, that you get the same results. There's no real difference. But something like acupuncture, we will pick up acupuncture in the next test, or in the next video. Sorry, folks.